Hello, welcome to your Chapter 2 lecture. This chapter will cover the basic unit of life, cells, and gene expression, as well as cell division, both meiosis and mitosis. You will find on your Blackboard site some supplemental resources for this module, both videos and practice worksheets. As always, if you are confused about any of the content at the end of the module, feel free to contact me to schedule a time so we can talk. Did you know that you are 100 trillion cells big? Did you know that the most abundant cell type in your body is not really a cell? Red blood cells are not true cells, and you will learn why later in the course. But their numbers in your body would have you believe that they were true cells. They are the most abundant, quote, cell in the body, as 25 trillion of your 100 trillion cells are red blood cells. There are 5 million red blood cells per cubic microliter of whole blood. That amount of cells would occupy the space of the period at the end of the sentences shown on this slide. But red blood cells aren't the only cells in your body, are they? Every cell in your body has a specific job to perform in order to keep your body in tip-top shape. The better word to use is homeostasis. Homeostasis means a stable internal environment. Let me explain. You want enough red blood cells, but not too many. You want some blood volume to make adequate blood pressure, but not too much nor too little. That all deals with homeostasis. Each cell in your body has a specific role to play, and each cell is the most basic form of life by exhibiting the 7 characteristics of life. They are organized. That means they have cell compartmentalization. They respond to stimuli. For example, cells respond to chemical signals. They maintain a constant environment. For example, each cell regulates its size. They reproduce through cell division. They grow and develop. Yes, there is such a thing as an immature versus mature cell. They adapt to their environment and acquire energy in one form to convert to a cellular form called ATP. These 7 characteristics of life are all accomplished by organelles. The word organelle means little organ. I'm sure you're excited to learn about these organelles, so let's get started. Human cells have some common characteristics. They have a barrier that separates the internal environment of the cell from the extracellular environment. This barrier is called a plasma membrane. The intracellular fluid is called cytosol, and scattered in this fluid are the cell's organelles and the nucleus. I will discuss the organelles in just a moment. The fluid outside of the cell is the extracellular fluid. As I mentioned previously, each cell in the body performs a specific function. When a group of cells perform a similar function, we call that group a tissue. Later on in this lecture, I will discuss how cells can become specialized to perform their specific function within our body through a process called determination and differentiation that are directed by gene expression. Some cells have a directionality to them. They can have a top surface, which is the apical side, a bottom surface, which is the basal side, and lateral sides. This directionality applies to epithelial cells, and this is discussed further in Chapter 4. Let's start with the plasma membrane. It's not an organelle, but it is a very important contributor to the cell's function. You see, it provides a selectively permeable barrier around the cell. That means some things can come in or out of the cell, and others can't, at least not easily. The plasma membrane is described as the fluid mosaic model. Scientists long ago did a freeze fracture method that allowed them to split apart the cell membrane into 2 halves. When they did this, they noticed that the membrane wasn't just a sea of phospholipids, a type of fat, but also there were proteins embedded within the phospholipids. Think of the proteins like an iceberg amidst the phospholipids. This membrane is much like the oil on top of an oil and vinegar salad dressing. The molecules that make up the membrane are made up of phospholipids. These lipids have unique chemistry. They have heads and tails. The heads are called polar because they have charged molecules. The tails are nonpolar because they do not have charges. I'll explain more on this during the next slide. In chemistry, we say like dissolves like, or like loves like. That means nonpolar molecules want to be next to nonpolar molecules, and polar molecules want to be by polar molecules. Keeping this in mind, let me tell you about water. Water is a polar molecule, and water is the most abundant molecule in our body. We are walking sacks of water. Well, an oily substance will not want to be next to a watery substance, as these are not like substances. So the polar heads of the phospholipids like water, and the nonpolar tails do not. There is water in the cell, that is called intracellular fluid, or ICF, and there is water out of the cell, that is called extracellular fluid, or ECF. The phospholipids will line up together with their heads oriented towards the watery compartment, either the ECF or ICF, with their tails pointed toward each other. This, of course, can only be accomplished by having a double-layered lipid membrane. Thus, the two parallel sheets of the phospholipid bilayer align so the tails of each sheet point toward each other. The cell membrane is also called a plasma membrane or plasmalemma. Take a moment to familiarize yourself with this picture from your book. A phospholipid has unique chemistry. Let's start with the glycerol backbone, shown at the end of this arrow. It is a 3-carbon chain. Each of these carbons have or had a hydroxyl group, which is an oxygen and a hydrogen atom. Phospholipids and glycolipids can be used for energy, but primarily they are used to generate cell membranes. These pictures are showing you a phospholipid structure. Your book describes it as a lollipop on 2 sticks. Notice that we see the glycerol backbone, highlighted in purple. Attached to the glycerol are 2 fatty acid chains. On the 3rd hydroxyl group of the glycerol, instead of another fatty acid, a phosphate group is attached. On the phosphate group can be attached more molecules, and these change depending on the type of phospholipid created. So here they are described simply as a, quote, non-lipid group. What you should realize is that the fatty acid chains are hydrophobic and want to avoid water, and the non-lipid groups attached to the phosphate are attracted to water. Therefore, phospholipids are amphiphilic or amphipathic, which means they have both water-loving and water-hating regions. A glycolipid is similar to a phospholipid, except that a carbohydrate is attached to the 3rd hydroxyl group of the glycerol backbone. These glycolipids are important as identification tags for our immune system to distinguish between self and non-self. As I mentioned previously, scientists describe the cell membrane as a fluid mosaic. Amidst the phospholipids are proteins that can migrate through the phospholipids, much like icebergs can float in the sea. In other words, these proteins are not stationary. They are not fixed in one place. The proteins that are found in the membrane can be integral, which means they span the entire length of the membrane. Or they can be peripheral, which means they are loosely associated with one side or the other of the membrane. Now I'd like to address the glycocalyx of the cell. The glycocalyx is thought of as the cell coat. It is formed when chains of carbohydrate molecules attach to integral proteins to form glycoproteins, or to the phospholipids to form glycolipids. The glycocalyx helps cells attach to each other, but also the carbohydrates are used as a type of identification so that the immune system can tell self from non-self. The way cells determine self from non-self will be discussed later on in the semester when we cover the immune system. Lastly, scattered throughout the phospholipids are cholesterol molecules, shown here at the end of this arrow. The cholesterol is like the cell's antifreeze. It helps to keep the membrane fluid-like, while at the same time giving the membrane more stability. Cholesterol is another type of lipid and helps to increase the hydrophobicity of the cell membrane. The phospholipids and the proteins associated with them give function to the cell membrane. The membrane acts as a barrier and separates intracellular fluid from extracellular fluid. It also provides protection to the substances within the cell from substances and forces outside of the cell. The proteins associated with the membrane can aid and regulate what substances come in and out of the cell. They can also act as receptors and bind to substances dissolved in the extracellular fluid. These substances are chemical signals, such as hormones, that can alter the function of the cell. Without the proteins, only small uncharged molecules, such as oxygen and carbon dioxide, can pass through the hydrophobic interior of the membrane. Other substances, like glucose and charged particles, cannot get through the membrane on their own. Rather, they will need an integral protein to create a pore, or channel, through the hydrophobic interior of the membrane. In some cases, the protein can physically bind to the molecules and transport it across the membrane. In the next few slides, I will discuss the modes of membrane transport. They include 3 broad categories: passive transport, active transport, and bulk transport, also called vesicular transport. Passive transport includes simple diffusion, osmosis, and facilitated diffusion. Active transport includes primary active transport and secondary active transport. Bulk transport includes endocytosis, which is further categorized as phagocytosis and pinocytosis, and lastly, exocytosis. I have provided a link on this slide. Cut and paste it into your web browser. There is no sound with the videos on the link, just a transcript you read as it plays. Allow the video to begin a bit and then click on Main. It will give you a menu to observe passive transport and active transport and all the subcategories I listed above. It places endocytosis within active transport, which is fine, but understand ATP is not used during this process.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Perhaps I should talk a bit more about simple diffusion, which is one type of passive transport. Diffusion is a process where a gradient exists across the barrier for a molecule. In this picture, we see that the red molecules are more abundant on the right side of the tube than the left. In the middle of the tube, we see a yellow membrane. The membrane has pores that are large enough to allow the molecules to pass through. With time, we see the molecules have moved from the high concentration toward the low until equilibrium has been reached. Molecules move because of the vibrations created in their bonds, a movement called Brownian motion. When equilibrium is reached, the molecules are still moving back and forth across the membrane, but there is no net gain nor loss of the molecules from either compartment. Rather, there is equal flux back and forth. As the previous slide discussed, some molecules can diffuse through the membrane and others require a protein to aid their transport through the hydrophobic core. An integral protein that has an internal pore will allow the extracellular fluid and intracellular fluid to flow back and forth through the membrane. Such pores can allow small, hydrophilic molecules to move across the membrane. The protein does not bind to these substances. Rather, they simply move through the pore using Brownian motion. And each protein is specific for the substance they allow to pass through. For example, there is a protein that creates a pore for potassium to diffuse out of the cell, and there is a separate protein that allows sodium to diffuse into the cell. Osmosis is another form of passive transport. In this picture, we see a yellow membrane separating two solutions. As with the previous example, the two solutions have unequal amounts of molecules. However, the molecules are too large to move through the membrane. If there is a concentration gradient with solutes, the red molecules shown here, and they cannot diffuse to reach equilibrium, then water will move. Osmosis is the water equivalent of diffusion. Water will move down its own concentration gradient toward a solution with higher concentration until equilibrium is reached. The equilibrium is when both solutions have equal number of molecules per unit of volume, or another way of saying this is the water will move until both solutions are of equal dilution. In a clinical setting, we are very concerned about the fluids we give our patients, particularly IV fluids. We must make sure the fluids have the same concentration as the body's fluids. If the fluids have more molecules than our body's fluids, like the right side of the first tube in the example, and the molecules can not move, then there will be a movement of water out of the cells and they will shrink or crenate. A fluid that is more concentrated than our body's fluids is called hyperosmotic. If we inject a fluid that is less concentrated than the body's fluid, let's say like the solution on the left of the first tube, then water will move into the cells and they will swell and possibly burst. Such a fluid is called hypoosmotic. The take-home message is this: if a cell membrane separates two fluids with unequal numbers or dilution of molecules, and if the molecules of solute cannot move, water will, and there will be a net gain or loss of water across the cell membrane. The left side of this picture is showing simple diffusion. The substance is diffusing across the cell membrane through an integral protein pore. The right side of the picture is showing facilitated diffusion, which is still passive transport in that no ATP is required. Facilitated transport and simple diffusion both have the same end result. At the end of the diffusion, the substance will be at equilibrium, where there is no net gain or loss, no net flux of the substance across the membrane. However, facilitated diffusion is very different from simple diffusion in the following ways. 1. The protein must bind to the substance to shuttle it across the membrane. And 2. There is a maximum rate this movement can occur. In simple diffusion, a protein does not chemically bind to the substance, and the substance moves with no limit on the rate. If there is a high concentration gradient, diffusion will occur more rapidly. With facilitated diffusion, the protein chemically interacts with the substance, and the rate of moving the substance is not dependent on the chemical gradient, but rather the number of protein carriers. Look, think of it like this. We're all going to go to a concert. At the end of the concert, we need to take a taxi home. We all want to diffuse home, but we need a shuttle of sorts. If there was only one taxi, we'd all eventually get to our homes, but it would take all night long. If there are more taxis, we could diffuse home a lot quicker. So the rate of facilitated diffusion is predicated upon the number of protein carriers. Active transport is another way to move molecules or ions across the cell membrane, but it is the complete antithesis of diffusion osmosis, and facilitated diffusion. Instead of particles moving down a concentration gradient, active transport is about molecules or ions moving against their gradient. That is to say, a gradient is being made, and this requires energy in the form of ATP. Active transport has two examples, primary and secondary. The pictures on this slide describe primary active transport. I will discuss secondary active transport on the next two slides. In this picture, you are seeing the activity of the sodium-potassium ATPase. It's named this to tell you it moves sodium and potassium, and ATPase means that it uses ATP in order to move the sodium and potassium. The pictures show the sodium and potassium being moved at different times, but really they are moved at the same time. The picture has separated them for clarity's sake. Potassium is being moved into the cell to create a large potassium gradient within the cell. Sodium is being moved out of the cell to create a large sodium gradient outside of the cell. Also, there are 3 sodium pushed out of the cell for every 2 potassium pushed into the cell. Again, ATP is required to build these chemical gradients. I want you to be mindful of the unequal amounts of sodium to potassium, as this unequal movement of particles will be of huge importance when we discuss muscle contraction for Unit 2. Now, something to think about. If this pump, this ATPase, is using energy to make a large extracellular supply of sodium and a large intracellular supply of potassium, how would these 2 ions diffuse if allowed? Do you agree that sodium would want to diffuse back into the cell if it could, and potassium would want to diffuse out of the cell if it could? Yes, they would. And what would they need in order to diffuse? They would need an integral protein to allow them passage back across the cell membrane. Such types of proteins that form pores for these ions are termed sodium leak channels because they allow sodium to diffuse back into the cell, and potassium leak channels because they allow the potassium to leak out of the cell. Now, I admit, it sounds like the action of the ATPase is all for naught because these leak channels just allow the ions to move right back from where they came. But actually, the leak channels are less abundant than the sodium-potassium ATPase, and the little amount of leaking that does occur is for good reason. I'll explain on the next slide. Secondary active transport is the type of transport across the membrane that utilizes those leak channels I told you about in the previous slide. And I'm going to focus on the sodium story. The leakiness of potassium is a story for Unit 2 and Unit 3 when we talk about excitable cells like muscle and neurons. So, in secondary active transport, the take-home message is that one substance moves down its concentration gradient. That is, it diffuses across the membrane. And the kinetic energy released from this movement allows the movement of another substance, but against its concentration gradient. Notice I said kinetic energy, not ATP. ATP is not used, not directly. Look, think of it this way. You and I are going to go skiing. We could use the chairlift to get up the mountain against the gradient of the mountain, but that uses energy, electricity. You and I want a more environmentally friendly way to ski. So I have an idea. I will take the chairlift up the mountain. We have 2 very long ropes. Once I get up the mountain, I'll throw one end of the rope down to you. We'll have a circle of ropes tied to us, and looped around each chairlift base, so that when I ski down the mountain, my momentum will pull you up the mountain. Then you ski down, and your momentum will pull me up. See, now we don't have to use the electricity from the chairlift. But notice we had to use it at least one time, right? Well, in primary active transport, the sodium-potassium ATPase uses the energy from ATP to establish sodium's extracellular gradient, remember? It starts the gradient. Once it's there, it can diffuse down its gradient, ski into the cell, and pull along another partner. In the examples above, watch how sodium diffuses into the cell, down its gradient, and it also pulls in other substances like amino acids or glucose or bicarbonate. Even though the other substances are also going into the cell, they are being pulled against their gradient into the cell. When both sodium and the other substances go in the same direction, we call this co-transport. When sodium and the other substance go in opposite direction, much like my skier analogy, this is called countertransport or antiport. Again, sodium is allowed to diffuse down its concentration gradient, while another substance is being pulled against its concentration gradient. And in antiport, the sodium and the other substance go in opposite direction.